Hello, my name is Araiza. I'm an exchange student from Mexico and my major is biotech engineering, and I'll be talking about antibodies. This is the contents to which I'll be talking about. First, I will talk about what are antibodies, as well as traditional polyclonal antibodies preparations. Then I'll talk about monoclonal antibodies and a technique for screening antibodies called phage display technology. Then I'll talk about some therapeutic applications, as well as their role in tumor immunology, as well as about tumor-associated antigens. Then I'll talk about the antigenicity of murine monoclonals, then about chimeric and humanized antibodies, and about antibody fragments, and I'll finish with some additional therapeutic applications. So antibodies are large Y-shaped proteins which recognize and will bind to a specific site called epitope of a foreign target, which in this case is called an antigen. Due to this property, they have a high impact in human healthcare, since they can be used for treatment of different infectious diseases, such as of specific microbial or viral pathogens, microbial toxins, and also snake and spider venoms. So now I'll talk about traditional polyclonal antibody preparations. In this case, we are talking about the process for obtaining it from an animal source. First, you immunize a large animal by injecting the antigen of interest. Then, once we get the desired concentration of the antibodies that detect this antigen, we collect samples, and then we purify them, which can be done through precipitation and then high-resolution chromatography, such as ion exchange. Then, after we have isolated the antibodies of interest, we add some stabilizers as well as some preservatives, and we adjust the potencies. And finally, we sterile filtrate them and add them to an aseptic filling, which then we can make a liquid product, or we can freeze dry it to make a powder product of antibodies. The downside of using antibodies from animal source is they can have induce unwanted side effects, which can be either not acute or also can be life-threatening. Therefore, antibodies which are obtained from human sources, such as immunoglobulins, are preferred due to they have less chances of having unwanted reactions. These immunoglobulin preparations can be obtained from individuals who have been immunized against the antigen of interest, or that have recovered from an infection from this antigen. And the process for obtaining this is very similar to the one used to obtain from animal sources. Monoclonal antibodies were discovered in 1970s when the Kohler and Milstein fused two different types of cells, such as a myeloma cell and a B lymphocyte cell. Those obtaining a cell called hybridoma, which has is stable cancerous and is able to produce the antibodies produced by the B lymphocyte which it was fused with. Therefore, it's able to produce monospecific, or also called monoclonal antibodies, without exhaustion. These monoclonal antibodies are able to bind to a specific region of a specific antigen. The process for obtaining hybridomas is first humanize the mouse with the antigen of interest. Then we remove the spleen of the mouse, which is rich with the B lymphocytes. Then these lymphocytes are fused with myeloma cells of mouse. Then we obtain these hybridomas, which are then separated from cells that were not fused, and then we screen for the hybridomas that produce the monoclonal antibodies of interest, and then we isolate them. This is an example of how hybridomas are made. So this is a technique for screening the antibody of interest. First, we create a gene library that includes our antibody of interest, and we insert them into the DNA of phages. Then we infect or we culture the phages along with E. coli to help their replication, creating mature phages which will present the antibody of interest on their coat. Then this will help isolate them through affinity screening, such as affinity chromatography. Then once we have immobilized the phages that contain the antibody of interest, we elute them and we recover the gene, which can then be inserted into a vector. These are some therapeutic applications of monoclonal antibodies, such as the induction of passive immunity against an antigen, also diagnostic imaging, as well as treatment of cancer, transplantation, and cardiovascular diseases. In order to use antibodies for in vivo diagnostic or therapeutic applications, we first need to identify the antigen, and then once we identify this antigen, we're able to raise monoclonal antibodies unique for this surface antigen. Antibodies can either be bare, or they can be conjugated with a radioisotope or a drug or toxin, depending on the purpose. Once an antibody binds to an antigen, it can have different responses, such as the cell might internalize the antibody-antigen complex. It can also shed from the membrane, or also nothing can happen. In tumor immunology, we can use antibodies that are able to bind to antigens that are expressed in the surface of cancerous cells. These are called tumor surface antigens. These cannot be expressed on healthy cells or are expressed in very low levels. Therefore, they can be highly specific for a cancer cell. Once this antigen is present, the immune system recognizes it and is able to destroy the transformed cell. These are some antibody strategies for using antibody for the detection or destruction of tumors. The first is using a monoclonal antibody conjugated with a radioisotope. When conjugated with a beta emitter isotope, it can be used to selectively deliver radioactivity to the tumor site. And when it's conjugated with a gamma emitter, it will make them congregate in the tumor site, we can then use a detection equipment of gamma rays in order to visualize the tumor. The most common isotope is technetium, which will emit enough energy to be detected, and since it has a short life, it will reduce the exposure of the patient with this radioactivity. We can also conjugate them with toxins, which will then, once the antibody will bind to the target cell, and once it's internalized, the toxin will induce its toxic effects, killing the tumor cells. We can also bind antibodies with drugs. Therefore, we can mediate the delivery of a certain drug to a tumor. We can either conjugate them to a chemotherapeutic drug, but this is not highly used because of the limited number of drug molecules which can be attached to an antibody molecule. Then another technique is used, which is called ADAPT, in which we bind an enzyme that will activate an inactive drug. Therefore, when we inject the antibody conjugated with this enzyme, it will congregate around the tumor. Then the drug is injected that is inactive, and therefore will only become active around the tumor site. Like we said before, tumor-associated antigens are antigens that are expressed on the outside of tumor cells. These different antigens are expressed in different types of cancers. When usually virus-induced cancers present the same antigens, even if they are different viruses or different type of cells infected by the same virus. Therefore, the antibodies can cross-react with different types of tumors. Also, oncofetal antigens are antigens that are presented during fetal development, but they are no longer expressed in later development. Therefore, when a cell is infected with cancer, it will re-express these antigens. Therefore, they can be used for biomarkers. It does have its complications because of the number of tumor-associated antigens that have been characterized to date. Also, some different cancer types can display different unique tumor-associated antigens in different patients. This usually happens with cancers caused by point mutations in a nucleotide sequence. They can also be expressed in lower levels in healthy cells, therefore they can be not highly specific. Also, the antibody, once it binds to the antigen, it can shed from the cell surface instead of being internalized. Also, these tumor-associated antigens can be expressed in only a certain cells of a tumor, not in all of them. Murine monoclonals, which are antibodies obtained from rats, can be used in immunogenicity treatments in human subjects, but it has its downsides since humans can have an immunogenic reaction, and therefore their effects can only be efficient in 1 or 2 doses before they become inactive. Therefore, the solution for this is the use of human monoclonal antibodies of human origin. How can we do this? Is by making antibody-producing human lymphocytes, making them immortal. This can either be achieved by Epstein-Barr virus infection, by fusing them with murine monoclonals, or by fusing with human lymphoblastoid cell lines. But this is very difficult due to the sources of antibody-producing cells, the unreliable methods of immortalization, and also we can't be sure about the stability and antibody-producing capacity of immortalized cells. Therefore, we can use DNA recombinant technology to generate hybrid antibody structures of murine and human antibodies. This can either be chimeric, which is the fusion of the constant region of the antibody with the variable region of the murine antibody, which is the one that binds to the antigen, therefore decreasing the immunogenic reactions and also the half-life of the antibody. Humanized antibodies is using human antibodies whose the binding region of the variable region of the antibody is exchanged with those of the murine antibody. We can also, due to some tumors are solid, therefore it's difficult for whole antibodies to penetrate them. Therefore, we can use antibody fragments, which are able to penetrate more easily solid tumors, but they have their downsides since they have a lower serum half-life, and they cannot initiate effector functions since they are missing the constant region of the antibody. Therefore, they are more better suited to be used for diagnostic imaging instead of therapeutic purposes. There are also other therapeutic applications aside from treatment of cancers, such as for imaging of vascular-related conditions, such as observing the region of the heart that has a myocardial infarction. We can also use it to visualize size of focused bacterial infections by using antibodies that bind to the surface of bacteria. We can also use them for the immunosuppression of patients which have autoimmune conditions. This by using antibodies that will bind to B or T lymphocyte specific surface antigens. This can also be used for the reduction of for reducing the chances of rejection of transplanted organs.